Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. Before we dive into this episode, we would like to take a moment and thank our ecosystem partners for their support, namely Domain Therapeutics, GPCR Therapeutics, Design Pharmaceuticals, Montana Molecular, and Orion Biotechnology. Mark your calendars for July 21st, as we will be hosting another free symposium in the ecosystem, this time on the molecular and structural insights of GPCR activation. Our program includes talks from Katerina Nemec, Alexei Serbo, and Polina Isankina. We're taking you on a structural and molecular journey to understand GPCR activation better. If you'd like to give a talk, we are still constructing the program, so please reach out by email at hello@drgpcr.com. We also set aside 2 hours for poster presentations and networking on a new space called Kumo Space for this symposium. There is no limit on the number of posters we can accommodate, and everyone is welcome to present a poster at our upcoming events, including the one on July 21st. We will not be selecting posters. You will not have to send in any posters. We only ask you to fill out the poster form so that we can capture your information. Part of that information is a 1-minute video abstract that we will share on YouTube. We'll also share it on our website so that people can see what you will be presenting about. You can also join us live for any of our events by marking your calendar and becoming a Dr. GPCR Ecosystem free site member. For more information and an updated schedule, you can go into the ecosystem. If you haven't noticed yet, we've added menu buttons at the top of our page for your convenience to help you navigate the ecosystem more effectively. And now, let's dive into this episode. Hello, everyone. This is Yamina from Dr. GPCR, and today I'm excited to have with me Dr. David Sykes. David, so exciting to have you on with me today.
Thank you very much. Yeah, Yamina, for inviting me.
Thank you so much for being here. I'm very excited to hear about your story, about what you've been doing. We talked about it a little bit before we hit record. So why don't we start at the beginning? Would you please introduce yourselves?
So yeah, I'm Dr. David Sykes. So I'm a Roche postdoctoral research fellow at the University of Nottingham. I'm studying cannabinoid receptors as part of that postdoctoral research fellowship, specifically looking at biosignaling. So yeah, I mean, my background is in GPCRs. I've worked on them for over 30 years. I started my career as a pharmacologist at the James Black Foundation in South London. And yeah, I was trained there initially in sort of radioligand binding assays. So the first GPCRs really that I experienced were the gastrin receptors, so CCKB and CCKA. So at the time, Sir James Black was very much interested in trying to discover blockers of CCKB receptors. So he was looking as almost like a follow-up to obviously the H2 antagonists and cimetidine. And so that was really my first experience of working on GPCRs. And yeah, that experience obviously was obviously instrumental really in me really getting to like this field as a whole. And obviously, once you've started working with GPCRs, you never stop working with GPCRs, I don't think.
Agreed. Did you always know that you wanted to be a scientist?
So, I mean, when I was young, I think my mum always thought that I would become a scientist. So she would find me in the kitchen, sort of mixing up certain potions in the kitchen. And I think she had an idea that I might become a chemist, I think. But obviously I was quite drawn towards the pharmacology side. So I remember sitting down when I was much younger and looking through like a university prospectus and just looking through the various courses that you could choose. and I was particularly drawn towards the pharmacology. So it just seemed quite exciting. Obviously, the study of drugs. Obviously, we all take drugs at some time or another, and that just seemed, it seemed like a really interesting sort of career to pursue. So that's really why I chose it as an undergraduate degree. So I studied pharmacology at the University of Leeds in Yorkshire in North England. And I'm actually a Lancastrian, so I'm from Manchester, but my name Sykes is actually a Yorkshire name, so I have some roots up in Yorkshire. So it didn't seem too far to really just cross over the Pennines there, and I really enjoyed my time up there. I was taught by some wonderful pharmacologists, actually. So Professor Ian Hughes, who I think was quite a famous sort of pharmacologist of the early 90s and a fellow at the British Pharmacological Society. And they were quite inspirational characters up there, the lecturers. They made the courses really fun and really enjoyable. And yeah, I didn't really, I had a choice. I was doing like a joint degree at the time. I was doing a combined physiology and pharmacology. And yeah, I decided in the end to go for the straight pharmacology, even though I was probably better at the physiology side of things. But it just, yeah, I was just thought that the pharmacology had a more in terms of a career, it seemed a more sort of tangible route, really. So that's really why I decided to go that direction.
Why why the joint degree? Where did the interest into physiology come from?
So, I mean, I'm quite strong. I was quite strong at biology when I was doing my sort of A levels, and so I found physiology quite easy, relatively,
Mhm.
in terms of the learning and the understanding. So yeah, pharmacology I found quite difficult relatively, and but it just was more an interesting subject to me. So I decided to take that route rather. I couldn't really see what job you would do with physiology, but I mean, obviously, I'm sure there are plenty of jobs in that area. But it just seemed more exciting the pharmacology side. So yeah, I decided to pick that topic.
When was the first time you went and worked in a lab and realized, actually, I love the lab? Obviously, the kitchen, your mom's kitchen, was potentially the first one, but that there's a difference between, being in a classroom and learning pharmacology from a textbook and actually witnessing its effects in the lab.
Yeah. So, I mean, in terms of pharmacology, we had some, I would say we had some quite traumatic experience actually. Some of the initial, I mean, some of the experiments that we used to do up there were quite interesting. So we would, I can remember one experiment. They were looking at like the LD50 of a drug on I've forgotten what the name of the organism is now, but it I don't know, it's almost like it was a really fast moving organism that moved through water.
Mhm.
And it was like, I don't know if it's like a water flea or something, but we gave them a certain drug. I can't remember what it was now, but then these things moved extremely fast then at that point, and then eventually they died. And yeah, I just remember being fascinated by the effects that this drug had on that population, and yeah, there's obviously animal experiments going on as well, and seeing for the first time the realizing just how serious it can be, and the responsibility you really have as a pharmacologist if you're working with animals or with tissues. So
Yeah.
it really sort of woke you up at that point, and that this is quite a serious topic.
And yet very fascinating. Over the weekend, we had to make a trip to the emergency room with my oldest. He had a stomach bug and he couldn't eat for, I think, almost 3 days. And the beauty of it all, if you can say, is that we get to the doctors and they give him a small little pill to put under his tongue, and it's an ion channel antagonist because I had to look it up there. And minutes later, the child is better. And I told
Yeah,
myself, wow, it's not a GPCR, but amazing how fast these drugs can
yeah,
act.
connect. And again, I one of the first drugs that I can remember having to be prescribed was actually cimetidine. So I think I'd been enjoying my undergraduate degree a bit too much up in Leeds, and probably drinking too much. And then I got back home over the summer, and I had to find myself developing some sort of ulcer. So I'd actually taken cimetidine as part of my treatment. And again, that is another wonder drug. It just works instantly and immediately you get better. So like this was before I joined the James Black Foundation. So obviously I had something to thank Sir James for when I arrived there, that he'd actually invented this sort of miraculous cure. And yeah, it's interesting. He was actually because he was originally a doctor himself. So yeah, when I was up there, if I did have any complaints, like I'd often go and sit in his office, and he could probably diagnose you and tell you what's wrong
Wow.
with you.
So how did you get into the Black Foundation? How did you
So
navigate
yeah, no,
that?
it's quite lucky really. I so my first experience of working in a lab was actually, or proper experience, was during the summer placements. So I'd been working up at the clinical oncology and radiotherapeutics unit in Cambridge. And my sister used to work there, and that's how I actually got my first job there. So she'd recommended that I come up there for the summer. So I had a student placement up there for a couple of summers while I was doing my degree. And then once I'd finished my degree, I applied for a job at Pfizer, and I didn't get that job. And I'd also applied for this job at the James Black Foundation. So I was delighted to get an interview. And I had to travel up from Manchester a couple of times for 2 interviews. And then I thought I was going to have to go up there again for a third interview. But by the time I actually got there on this third interview, the first thing they did was offer me the job as soon as I got in, which was quite nice because but by this point, I was, yeah, I was obviously, yeah, it was quite a sort of a strain to have to go up there twice, thinking, God, well, do I really have to go through a third interview? But yeah, so that worked out quite well, really, and yeah, I mean, I then once I'd got that position, as I say, I think I got it on like a 3-2. So I think there was some debate whether or not they chose someone who had slightly more experience, but they decided to go for me in the end. And yeah, I was obviously very grateful to my first boss there. So that was Dr. Elaine Harper. So she was the first person who trained me in sort of techniques of radioligand binding. So that's obviously something that I continued throughout my career, really. I've been working with radioligands, looking at the kinetics of binding, really, of drugs to receptors, to GPCRs. And yeah, that was a really good experience. And I moved sort of around the foundation. So I started off in the radioligand binding, but then I moved over to the organ baths to really do sorts of functional pharmacology. I was working there with another good scientist, Dr. Gillian Watt, who's now quite a senior member of Pharmaron. And yeah, again, I learned a lot from her. She was sort of an expert in organ bath experiments, very good at understanding the maths behind the pharmacology. So I learned all about the equations, and that really stood me in good stead. And yeah, I then did a master's while I was there. So the head of pharmacology there was a guy called Nigel Shankly, and he'd actually been involved in setting up the original operational model of pharmacology with Paul Leff. And well, I remember one day in his office, we were just sort of talking about careers, I said to him, I feel, Nigel, a bit like a jack of all trades, master of none at this point, because I was still quite a relatively young scientist there, and I kind of moved around a lot, studying a lot of different things. So he said, "Oh, okay." He said, "Master of none." So he said, "Well, why don't you do a part-time master's then?" And he said, "Yeah." So I was obviously delighted, but he said, "Yeah, on the condition that we'll only pay for it if you pass it." So,
Okay.
so, so it was obviously a little bit of pressure then for me to make sure that I passed this masters. But that was a really good experience. So I had half a day off a week, which I travelled to King's College in London to study for my masters. So I did a masters in molecular biology and biochemistry, and um, yeah, I then transitioned then from the sort of the organ bath experiments into a new lab that was being set up in the foundation. So we recruited a lady called Carol Austin, who's now at Eurofins, and she really taught me all about different types of cell culture, and obviously I learned some new skills in molecular biology at that point. And that was really my first taste of doing sort of cell-based and plate-based assays as well. So I remember running sort of cyclic AMP assays in that lab. And I think we were one of the first groups to sort of beta test the Lance cyclic AMP assays from PerkinElmer. So that was a really fantastic experience. And yeah, I mean, I think that's been one of the keys to my career, really, is to maybe keep hopping around within pharmacology and really getting a taste for different techniques. And that's something that I've carried on now as well. So after I did my PhD with Stephen Charlton at Nottingham. So this is only quite recently. So although I've been a pharmacologist for probably over 20 years at that point, I then decided after Novartis had closed down and I joined Nottingham, I decided to do a part-time PhD. And again, in terms of trying to learn new techniques, one of the things that we decided to do was to try and get away from radioligands. So obviously, I've been sort of profiling lots of different compounds to study their kinetics using radioligands in the past. But we decided or made a choice to actually move away from them and then start looking at fluorescent ligands and these new techniques of FRET and BRET. Yeah. And so as part of that PhD, I developed a FRET-based kinetics competition assay, and we were profiling then. Initially, we sort of had it as just a test case. We thought this would just be a control set of antipsychotics where we would reproduce the literature and show that there was this relationship between the off rates of these ligands and extrapyramidal side effects. But actually, it turned out more interesting than that. So we profiled these compounds using these fluorescent tracers. And what we discovered, actually, there wasn't really a relationship between the off rates of these antipsychotics, but it was more the on rates that were driving the extrapyramidal side effects. So Stephen Charlton has been working with another famous Belgian pharmacologist, Georges Verquelin. So he's obviously quite famous for studying kinetics and the hemi-equilibrium of antagonist binding. And he's also developed a number of models, including this rebinding model, to better describe the actions of drugs binding to receptors in sort of non-stirred conditions. So where you've got this model that they developed, it became quite apparent that actually it was probably more the on rates of these antipsychotics that were driving these extrapyramidal side effects, and so we sort of expanded this rebinding theory, and that was probably one of the most the satisfying point of the PhD in many ways, and sort of the high point. But I did go on and study again, developing more of these FRET-based assays using different fluorescent ligands, this time for 5-HT2A receptors as well. And again, that's led to some interesting discoveries around some of the antagonists. So citindolol turned out to be a very slowly dissociating ligand at that receptor, and that's something, again, that wasn't known at that point. So I think that's something about kinetics, which is always quite interesting, is that if you profile a series of ligands, you will always find something interesting that helps you sort of understand the mode of action of those compounds. And yeah, I mean, I've got to say, in terms of, like, I spent probably the first 20 years of my career just measuring the affinities of compounds for receptors. But it's once you realize that drugs can have these identical affinities but very different kinetics, that you realize that actually we should be always trying to measure the kinetics of those compounds. If you can measure the affinity, then there's a good chance that you can probably measure the kinetics. So it just requires a little bit more effort, but ultimately it's always proved a worthwhile exercise. And we've learned so much about the clinical compounds that we've been developing. So I've been very lucky enough to work on a number of clinical compounds while I was at Novartis. So we developed a CRTH2 antagonist. So, and again, we didn't know why this compound was better than our original compounds until we started to study the kinetics of that compound. And it became apparent again that this compound had a much slower dissociation rate. So that was actually one of my jobs at Novartis, was to profile our compounds versus competitor compounds. And so we were essentially always making stories around the kinetics of these compounds to help promote them, really. And yeah, I looked at glycopyrrolate, which was one of our compounds to treat COPD. And again, that's got quite a slow off rate relative to compounds like tiotropium, which were more established for treatment of COPD. But that compound, again, you find that compound has a very slow off rate, which means that it's got a slower on rate. So there's always, I mean, I know it's a spin, but it's kind of, there is always something to tell about your drug if you can understand those properties. And it helps you really explain better why your compound might have some advantages over another compound.
And then having the tools to be able to measure these is absolutely crucial, because as you mentioned, if you can't measure, if you can only measure affinity, that's great, but that's definitely not enough. And I think maybe it's just me, but the thought of, I would have never guessed or made an educated guess to think about the on rate being the one responsible for the side effects. I would have said, oh, maybe it's the off rate, which is a super interesting finding.
I mean, I think this will prove to be quite pivotal in terms of you will see this more often. I think that now on rate will become more important. So it's very clear. I mean, I started to look at data on from PET imaging. So if you look at that data, you see that these drugs, when they're administered, they're lasting for over 24 hours in the brain, many of them. And again, this rebinding effect, I think, is what's driving the duration of action of those compounds in the brain. And it can lead to some quite interesting effects in terms of the selectivity of those compounds at different receptors. So if one of the receptors that you're targeting, if that drug has a much faster on rate, then the duration of its effect on that receptor is going to be potentially much longer due to this rebinding phenomenon. And it's really fascinating. I'm sure that we'll see advances in the treatment of some of these CNS diseases once people start to really try and optimize maybe the on rates of these compounds as opposed to the off rate.
Let's take a small step back. You mentioned Novartis. How did you get into Novartis, and what made you go from academia to industry and then back to academia?
Yeah, I mean, so when the founder, it became clear. So Sir James retired, and um, and it wasn't long after then that they basically pulled the funding. So the foundation was funded by Johnson and Johnson.
Mhm.
So we spent a very long time working on gastrin receptors. I mean, a phenomenal amount of time, which I guess nowadays you wouldn't do that. You would have always had multiple projects running at the same time, but we had a single project essentially that ran probably for over 10 years. But we did start to look at other receptors. So we started to look at the H4 receptor, and which wasn't cloned at the time. So we were actually working in the wrong system. We were actually studying histamine receptors. But yeah, so we had another program on PTH receptors as well, which produced some interesting results. But again, we just couldn't find any other clinical sort of molecules, and I think that's why we lost the funding. So once that was lost, I then started to think about, well, what should I be doing really? Should I just hang around and wait for the redundancy package, which would have been probably quite good? But I thought I hated the idea of seeing the foundation just close around me. So I made this step that I was gonna move basically, because I just didn't want to see that happen, because I thought it would just be really sad to just see it close. So I started looking around for jobs. And one of my friends actually that I used to sit next to at the foundation, Dr. Elliot Lilly, he'd already made his way down to Novartis in Horsham. So I was I'd obviously heard he was having a good time down there. So when I saw a job come up, I applied for that position. And yeah, I was interviewed by Mark Dowling and Stephen Charlton, who obviously um, had published their paper in the BJP on the binding kinetics of the muscarinic antagonists. So Mark was obviously very much involved in setting up those kinetic assays. And I was just really inspired at the interview, just speaking to them and just having read the paper on the way up to the interview as well. I was like, just found it fascinating, really. I was almost like, wow, I've been measuring affinities for this long, when really it doesn't really mean very much. I mean, it tells you obviously about effective concentration, but it just doesn't tell you about how the drugs are working, really. So once I got there, and my job was actually initially described as being looking into the mode of action of compounds binding to GPCR. So and trying to really understand the clinical mode of action of some of the compounds that they had in development. So I looked at beta 2 agonists, muscarinic antagonists, and also the CRTH2 antagonists while I was there. And that was, I felt quite in a privileged position at that point. Although eventually they did get me working on just the normal sort of drug discovery programs, and I was screening and developing assays,
Yeah.
just like everyone else there. I mean, he's got very much an academic sort of bent towards, obviously, do your job, but if you have some spare time, then let's look at something more interesting as well. So I moved to Nottingham with another scientist, Dr. Liz Rosethorn, who's at Signature now, but she produced a very interesting paper on bias ligands and bias signaling at the H4 receptor while she was at Novartis. So that was a completely, I mean, I don't think that we didn't have a H4 program running, but she was profiling some of the ligands, looking at new technologies, and they just came across this interesting finding, and they just kept pursuing it, and eventually they produced a really nice paper in molecular pharmacology, which I'd recommend everybody read, actually, on the bias signaling of this J&J molecule. So that's what I would call like a real bias molecule. So it has beta arrestin signaling, but it has no G protein signaling. So I think that's one of the rarer molecules, really, and that's a fascinating paper. And yeah, we tried to do a little bit more work on that when we arrived here as well. So I've just read an interesting article by Bob Lefkowitz talking about in Nature about how Septerna are setting up these screening assays, looking at the effector proteins and trying to really understand how drugs are binding in the presence of these different effectors. And that's something we were interested in too, because we'd kind of realized that probably this bias signaling, it obviously is something very strongly to do with the binding of the arrestin and the conformation of the receptor in the presence of arrestin. So we tried to set up sort of conformational screening assays using some of the techniques that have been developed in Nottingham. And I'm also currently in my new position, I'm pursuing this idea of sort of precision pharmacology, if you like, and actually trying to again engineer systems so that we've got G proteins, arrestin, and we've just had Carsten Hoffman down here actually, who's been talking to us about GRKs, and that's sort of stimulated a few new ideas, because obviously people talk a lot about arrestin, but maybe less is known really about the preceding steps and GRKs. And I think, you know, that this idea maybe that you can have more precision pharmacology in the presence of effector proteins, because all the radioligand binding assays that we run, that we always try and drop the receptor into sort of a ground state, don't we, with that, and remove the effector protein.
Yeah.
So I think that's really going to be an interesting sort of avenue of research moving forward for drug discovery. He also picked up on sort of allosteric modulators as well as being another sort of area maybe where we'd see an increase in the number of drugs. I did work on an allosteric modulator project actually when I was at Novartis, and that proved actually quite difficult. So I think that some of the sort of the properties that make them unique. So obviously these ligands, they did bind uniquely to the human receptor, and obviously, yeah, once you start moving to other species, it's then very difficult to then see these effects in those other species, which then makes development, I'd say, quite tricky. So, so there's a lot to think about. It's a very interesting article, actually. Yeah.
How interesting. It's funny because I was playing around and testing ChatGPT, and I did ask it what was the top 3 advancements in the GPCR field in the last 5 and 10 years. And functional selectivity was number 2. Number 1 was cryo-EM. And I can't remember if it was the 5 or 10 year mark, but it was protein complexes, signaling complexes. Yeah. And I thought it was pretty accurate of a description when I went over that.
Yeah, no, I think that's going to be fascinating. I mean, that was supposed to be my PhD originally, was to try and connect the effector proteins, and I did do a little bit. So on H4, we did find some interesting, actually. We haven't published this yet, but what we found was those bias ligands from the J&J. When you connect the arrestin protein to the receptor, the off rates of the ligands, of the bias ligands, those are the ones that changed the most. So it does appear like there might be some link there between effector protein binding and the conformation then that these ligands were recognizing. So yeah, I guess it was always interesting. You could always see a little bit of G protein binding in your binding assays if you leave the salts out, and if you tailor the assay that way. But obviously to get complete effector bindings is not possible in just a standard membrane preparation. But the lab that I'm working in, so with Dimitri Viprinsev, I mean, it's very interesting. He's obviously a fantastic structural biologist. he can purify sort of any protein. And that's what I've been doing more recently. So I've been learning to solubilize GPCRs. We've set up a number of interesting techniques actually just over the three or more years that I've been here, and we've been working on a technique called thermobret. So this is something that we've just published on in bioarchives, and something that, well, the full paper should come out soon, but this was developed by actually a postdoc who visited from Australia and who's now gone back to the Florey Institute. Very talented guy, Brad Hoare, and he developed this idea of tagging the receptors with a thermostabilized nanolook, essentially, which doesn't unfold as you increase the temperature. And then in the presence of ligands, you can obviously stabilize these receptors and observe a change in the TM of the receptor. And so you can then start, essentially using that as a tool to pull out binders for your receptors. And that's been quite an interesting technique to work on. And more recently, we've been looking at trying to develop sort of membrane-based biosensors. So we've been using the GI case system from Gunnar Schultz's lab, and we've basically again been able to prepare membranes from that system and then observe decoupling of the G protein in the presence of various agonists, or see it an increase in the Brett signal in the presence of inverse agonists. So it's quite a high throughput technique now. You can put these membranes in the freezer. You can profile, hundreds of ligands if you need to. We've been able to do that for GI receptors, but GS proved a little bit more tricky. But that's something that I'm sort of involved in at the moment and that we'll pursue. So we had a very talented master's student, Morgan Scott Dennis. She's actually just written a paper on this that will be published hopefully published in one of the frontier journals.
Mm-hmm. Wow, you're keeping very busy. You mentioned cannabinoid receptors in the beginning of the conversation, and I do ask this from everyone. What is your favorite GPCR?
So, I mean, obviously, I'd have to say it is probably the cannabinoid receptor. I mean, I'd say I have a number of favorites. I'd say the beta-2 receptor is obviously standard favorite. I've used that many times as a sort of a control. That's my kind of standard receptor, because I think we understand how it works so well that it's just a very useful system to profile things against. And I use that as a positive control for if I develop a new system. I'd say, the cannabinoids currently are my favorite GPCR. So we're trying to study biosignaling, and so we've set up a number of different systems in order to do that effectively. So we have obviously effector coupling from the mini-G. We have effector coupling from beta-arrestin. We're trying to set up some other assays. So these are the signaling-based assays, but we're also trying to set up binding assays, which also incorporate the effector proteins as well. And that's proved to be very interesting. So I wouldn't say I was a huge believer in bias before I started, apart from maybe that H4 story I told you about, but I've now seen it firsthand. And I've seen it in not just on CB receptors, but I've also seen it at the beta-2 receptor. So I'm starting to think that, yes, it's definitely achievable and it's real.
Yeah.
But again, I think it is this idea of maybe the precision pharmacology. It's very easy to sort of generate these numbers, but again, maybe trying to understand what that bias means actually in terms of sort of a clinical effects of these compounds. So I'm pretty certain, say with the beta-2 system, that some of these compounds, potentially that bias that we're observing could could actually be playing an effect in that clinical response that's been observed for some of these compounds. So that's interesting. In terms of CB receptors, there's a lot that's not known really in terms of what disease areas to treat. So I know Roche has they're hoping to treat macular degeneration.
Mhm.
And that obviously involves like a local application of a CB2 agonist to the eye. So that's somewhere again where maybe, if you think about sort of beta 2 agonists and M3 antagonists in the lung, you don't necessarily have to worry so much about the PK then of those molecules. So because it would have been local application. So
Yeah.
I think one of the things that's clouding the field as well has been probably the lack of selectivity of the compounds. So we find, having profiled them in various assays, that they do tend to hit CB1 receptors as well as CB2 receptors. And occasionally you see some weird effects like things that are known to be inverse agonists at CB2. They can also be agonists at CB1. So I would imagine that a lot of the animal work as well, I know from talking to Uwe Grether at Roche, the guy who heads those programs, he often tells me that the pharmacology in the different animal species can be very different. So again, you can get antagonists in one species, but agonists in another, and obviously that's complicating the preclinical work. Yeah, and as I say, that first generation of compounds that's out there is probably just probably not sufficiently selective enough. So I think they've gone back, and Roche have had a program for many years, so they've been trying to develop more selective agents. So it's probably now is the right time really for where we maybe see an advancement in that field. I know there's debate about mainly CB1s found in the brain, but people have started to maybe consider that CB2 receptors can also be upregulated in certain conditions in the brain. So I think Alzheimer's disease potentially might have a role in the formation of the plaques, and there's some suggestions that it might have effects in a sort of reward based situation. So in terms of like drug addiction, so I think it could be still quite an exciting area to be working in.
I think it's very tricky because I do believe that there is bias. There's biased molecules, but I think context matters. And if you have the receptor expressed in different tissues, then context changes. And add on top of that a disease state, and then context just gets messed up more than a physiological context. So I think it's very, very difficult, and you have to be lucky, or you have to have, I don't know, I think the stars have to be aligned at some point in order to really hit the receptor at the right place at the right time to generate the correct effect that you need to have.
I like Terry's. I mean, Terry's definition, Terry Kanaka, is very clear, isn't it? That the drug must produce a different active conformation, and I think that's something that we've got to bear in mind. And I think that's why the conformation screening is potentially so important, because you're going to find that out essentially if that drug is really acting through a different conformation.
No, agreed. I was wondering, and this might be a very naive question. You mentioned measuring binding kinetics, and then the K-on rate matters in the example that you had mentioned. How does that translate into signaling bias, or does it have an effect on signaling?
Signaling bias.
Just on signaling. I was wondering if you have a molecule that binds very quickly, or there is a difference between different molecules when it comes to the K-on. Are there any examples in the literature?
I can't think of anything that springs to mind, but I would imagine, now this is maybe is a bit of a guess, but I think with sort of the on rate, obviously it means that molecule is going to when it leaves the receptor, it's got a very good chance of going back on the receptor. So you would imagine the drugs that with a very high K-on values, they may get internalized with the receptors potentially, because it's often related to the lipophilicity of the compounds. So when we talk about this compartmentalized signaling, probably the drugs that are going into the cells, this could be some of the drugs with the very high on rates. So when I was looking at spiperone binding, this was quite interesting. So we were labeling the receptors with a fluorescent spiperone, and you could only dissociate half of that molecule on the live cells afterwards, because essentially it would get taken up into the cells and taken into compartments where your antagonist could then no longer reach that. So I think that, again, that sort of, in the lung, the on rate is very important. So I think that what gives you the duration of action. So again, the on rate's related to lipophilicity, and so it means that drug is going to hang around in the lungs, and this effect of rebinding is going to occur quite readily. So there was always this phenomenon of sort of reassertion, where you could essentially apply like an antagonist, but then if you washed it away, the agonist would remain, and it would then reactivate the receptor long after you got rid of the antagonist. So again, I think this sort of, the on rate can have a tremendous effect on the pharmacology of your drugs.
Phenomenal. I also ask this from everyone, and I'm wondering, well, I think the answer is always yes, but do you think GPCRs are still good drug targets?
Well, no, I do. I mean, I think it is interesting. I mean, they are still probably the most widely drugged target, aren't they? So, I'm very interested in orphan receptors. So we again, from this idea that we could solubilize orphan receptors, really maybe start looking again at DEL libraries. So again, we've been thinking about this too. Obviously, we're quite good now at solubilizing receptors. We do have some DEL libraries in the freezer, and this is something that we're looking to explore next, really. And I think, just this idea that you can have all these molecules in the same test tube, and you need very small concentrations of these things, actually. So there's very few copies that are actually in these libraries. But then obviously once they stick to your receptor, you can wash away all the free, and you're just left with these compounds essentially that bind to your target. And then you use the DNA tags to then uncover what that molecule is. I mean, it's fascinating, and I'm sure that this again is going to be quite revolutionary for GPCRs. And I think again that's something that Bob Lefkowitz was talking about again in his Nature interview. And I think the idea again that this will pull out sort of allosteric modulators as well. I think that's fascinating. And whether or not allosteric modulators will make good drugs, I think this is the key question now. I mean, they tend to be quite low affinity, or a lot of the ones that I've studied are. So, like, actually, probably the developability of those molecules is actually quite difficult, I think. But it's another challenge, isn't it? I guess for GPCR pharmacology. But
exactly, more work for many more postdoc students, professors, and people in industry as well.
Yeah. I mean, I think I've heard that sort of drugging receptors now is not the problem. So finding molecules that bind your receptor is no longer the problem. Finding drugs that activate your receptor is not the problem. But actually, the problem is finding out what those receptors actually do. So I think that's probably the problem with most of the orphan receptors. We just don't really know what they do. And I was thinking about this in terms of like clinical trials. Like, should we be running sort of like more clinical trials, but on smaller groups? So once you've got a molecule, really trying to find out which disease is that molecule actually best in, because I imagine there's been plenty of molecules developed throughout the years that started off with an indication, but that's not the indication that they've eventually ended up in. It's so it's I think there's always value in discovering a good molecule for a GPCR. Maybe again the kinetics is something again that people should keep looking at, because that's always helped us differentiate molecules. So although your molecule fails, if it's got the wrong kinetics, then maybe that could be one of the explanations why you're not getting the effect in the disease model that you might predict.
Yeah.
I don't think many people are really studying the kinetics. People are still just studying selectivity based on affinity.
Probably. And I'm wondering how many times people actually do binding as a first step into understanding the molecule, other than keeping the binding assay as a secondary assay and doing regular, what I would call regular endpoint assays, and then saying, oh, well, it generates this much cyclic AMP. Let's see how much binding, how does it bind? But the kinetic aspect of both assays is not being taken into consideration that many times.
Yeah, no, I totally agree. And there are some fascinating companies out there actually now, aren't there, that actually try and study this whole thing in combination, and from
Yeah.
functional data, they'll try and predict the kinetics of binding.
Yeah.
It's obviously
Much,
quite a fascinating
much,
subject.
much more to do. You mentioned the DEL, well, the library, the DNA encoded library, and also the ability to purify receptors, not necessarily with orphan receptors, but with receptors that are known. If you were to stabilize a receptor in a specific conformation and then use it to screen, to increase the possibility of getting molecules that
Yeah,
are, agonist, antagonist, or
yeah, no, that's a brilliant idea, and it's something again that we hope that we're going to be able to do with these different effector proteins. So that's something we haven't yet been able to test, whether or not in the solubilized state, whether the effector proteins are still able to bind. Although one of the PhD students actually has, I think she has shown it. So she has been looking at mini G binding in the presence of solubilized receptors, and that assay does still work. Yeah, so
it's
that's something.
amazing.
I think that's on bioarchives actually. So, but this is, I mean, I think it's a fascinating area. Again, I haven't purified any proteins myself yet. That's obviously something Dimitri's again a specialist in, and yeah, it's just fascinating really to see what can be done now in a test tube.
Yeah. No, agreed. Agreed. Especially if you can translate the test tube into a plate to make it faster and more high throughput.
Yeah, I
That's
mean, so I think we're trying. I mean, this is something I'm really interested in. So I'm always trying to miniaturize the assays and make them high throughput because of my background. It just seems natural thing to do. So I'm obviously it saves a lot of money as well. So if you can
yeah.
miniaturize things, that was really the idea behind the membrane-based biosensor, was just to try and reduce the cost, and reduce the sort of the variability as well, and just reduce the amount of tissue culture if you can. So I'm always trying to, if I can get something out of the freezer, which is often what we do for binding assays, then that just saves you so much time. So.
Yes. Yes. It allows you to have a life of your own and not be a slave to your cells growing.
Yeah.
I do remember my time in the lab being really enslaved by the hex cells. If they grew faster,
Yeah.
then you had to be in the lab sooner to run the assay. If they didn't grow, then you didn't have enough cells to run the assay, and it was all the time this constant fluctuation.
Yeah, I mean, I struggle sometimes with this myself now. So we have some signaling assays that are currently running, and this is the only way the assays can be done. So
yes,
but
of course, of course. We'll respect
but,
the cells, but still can be
but
really convenient.
it's good. It is good. I mean, I think this period of my career is this sort of work-life balance. I mean, I think this is an interesting sort of point you draw on actually, and something the demands maybe in academia are probably just as high as they are um, in
Industry.
industry, if not higher in some ways, because you're really struggling for your next sort of grant opportunities. And in order to do that, you've got to keep generating useful data. So yeah, if we were talking, I don't know if you'll get onto this, but some advice for younger scientists would be, really to, make sure that, make sure that you get a good mentor as well, and just really, really sort of, look to sort of get advice really on that sort of the work-life balance really, because it's something maybe your immediate boss can't give you, so you really do need maybe someone on the outside, just to give you that little bit of advice on how to balance your role in academia. It's not something that I have, but I think that's something that would have been quite useful in my career. Maybe it's not something most people have. I think maybe more the younger people are seeking those out, because it's a program now that the universities are trying to put in place early on in their careers, and often the PhD students can either act as mentors, or they'll be mentors for the PhD students themselves.
No, I think it's really important to be able to have someone to talk to who's not directly involved in your career, because then that person can offer you perspective.
Yeah.
And then there is also the separation of interest as well. And the mentor who is not necessarily directly involved in your research can be a little bit more objective, I want to say.
Yeah.
Any other advice for young scientists? Because you led us into the inspiring young scientists. No, that's perfect. That's exactly where I was going.
So I think it's important to keep. So one of the things about my early career was, I was given a project actually to work on by myself at the foundation. So this was something that I don't necessarily think I'd been seeking out, but maybe Sir James had seen some enthusiasm in me. He thought, well, there isn't really enough hands to run this project, so we'll give it to David to do. So I ran this program against the RAGE receptor, and so it's the receptor for advanced glycation end products. It's not a GPCR, and actually this was quite a problematic project. So I'd started with I read up on the literature, but then I chose to go down a certain path to attack the problem. And so I developed these ELISA assays, and I was getting beautiful standard curves, but I couldn't get any stimulation with any of these ligands that I was trying. And so it turned out that actually the cell line that I'd chosen didn't have the receptor that so it didn't have the receptor for the positive control. So my positive control never worked. So I was at that point, you're starting to think, well, is it me or is it the system? So I think I've really understood the importance of positive and negative controls. I think this is quite important for young scientists. So but failure, that was probably, I failed many times in that project, but I learned so much in that project. I learned an absolute ton. Looking back, I learned how to grow all the different cell lines, because I was just moving between different systems. I read so much literature. In the end, it turned out that our problem was actually we were keeping these materials too sterile. So this receptor, basically a lot of the literature was false. Basically, it was all down to endotoxin contamination, and it was binding to these toll receptors on the cell. And so I learned a hell of a lot during that period, actually. So failure could be a really good thing, I think, as long as you're resilient and you can keep going. And I think, if you do attack a problem, attack it from many different ways, because it's like rolling a dice. You will get lucky, because just by chance, one of the paths you take will lead to something positive. Whereas I think if you attack a problem from just one angle, if you're unlucky, you will get quite disheartened quite quickly. So always attack a problem from multiple angles, and that's something I always do now. I always have different ways of attacking a science problem. other bits of advice, yeah, read as much as you can, basically. So the more you read, the more interested you'll become in the subject in general. So the more I read, I'd say up until my 30s, like my early 30s, I didn't read as much as I should have done.
Mhm.
And it was only at that point that I started to really take a greater interest in the subject, GPCRs as a whole. And I think that's something that you should do early on. So when I did my PhD, that was I found that quite relatively simple to sort of sit down there and read the literature, and it was a real sort of luxury in many ways at that point, because you have that time in that initial phase, and that's something all PhD students should be doing now if they're just starting, is uh, really diving into the literature and planning ahead essentially for what you hope to achieve based on the literature and how you're going to do it. So, yeah.
No, I like that. And I think one thing I'd like to add is talk to people and figure,
Yeah.
have the conversations. I mean, working with the cell line that doesn't have the receptor is it's the most basic error, and I've been there, and I've
Yeah.
had colleagues who've been trying to measure the cell surface expression of a protein only to realize that the DNA didn't have the transmembrane domain in it. So the protein was expressed, but it got secreted. And every time they would getting ready to do a flow staining, they would just flush out the protein in the cell culture media. And having the conversation with someone, well, actually two types of people: someone who knows nothing about your project, because then they can ask the most naive questions and make you think. And
Yeah.
the other thing is someone who is very good and is very knowledgeable around that project, because then again, they can guide you to the right paper, to the right people, and
Yeah.
and that helps concentrate the chances of success.
Yeah, it's very important. Thinking about that, it's very important to understand every component of your system. So often you inherit an assay, sometimes, don't you? And
Yeah.
if you don't actually understand all the components and what they're there for,
Yeah.
that can be quite problematic for you.
Yes.
If you're looking at something like, I remember studying S1P receptors, and I didn't have any sodium orthovanadate in there to prevent this thing from being broken down,
Mhm.
and you know I couldn't quite reproduce the results of the
Yeah.
person that had been working on it prior. So, it's really important, the fine details. I mean, I am
Yeah.
quite a details oriented person, but yeah, really understanding everything that's
Yeah.
inside your protocol.
Agreed. David, last question. Top three aha moments that you had as a scientist and that shaped your trajectory.
Okay, so I was thinking about this, and one of my sort of early memories of sitting in meetings at the James Black Foundation. So you often like hear a really complicated story from someone being presented, and you'd be like, they'd come up with some sort of question like, how do I solve this problem? And obviously, you've heard this very complex story, and you're thinking of the most complex solutions. And I always remember, like Sir James, he would just calmly come out with like the most simple sort of question. It would be the most simple solution that you'd ever heard of, and you'd be like, I mean, how did you even think of that? It would always be. I would just yeah, I would almost be sat there thinking, how do I think like that? So I think, yeah, again, just thinking the most simple ideas are often the best. It doesn't have to be like a complicated idea, does
Yeah.
it really? And so that's something that I learn. I always, if I think about a problem now, I'll always try and look for the simplest idea, really, to attack the problem. Um, Another aha moment. I suppose the rebinding as part of my PhD, and just the whole story around the
Yeah.
kinetics of binding. Really, I mean that again was for me was a huge moment, just to realize, wow, you can actually tell so much more about these molecules. So I remember when I was at the foundation, we were observing effects of these competitor compounds, and they would like suppress the maximal responses of the tissues.
Mm.
And retrospectively, I don't think we ever really discussed this, but it was sort of seen that these compounds were in some way inferior because they weren't producing like a sort of almost competitive, or what you would expect of a competitive antagonist. You weren't getting the rightward shift, you were getting suppression of the maximum response. So they sort of painted these as being inferior, when in fact, I actually think, maybe looking back now I understand what was happening, that they were probably the superior compounds. So I think that was another sort of eureka moment. And I don't know, a third one. I think, I mean, obviously you must always look to sort of keep reinventing yourself, really, and I think that's quite important. So you should always be looking to study new technologies and just stay up to date on various topics. So I think studying Fret and Brett is obviously, that's going to extend my career, hopefully. And I think that's just a good advice in general, really.
And I think that's a theme that followed you throughout your career, is really spending time and learning different technologies, different techniques, while staying with pharmacology and GPCRs.
I think, I mean, I have worked a little bit on some non-GPCR projects just recently for the company Z7 Bio, which is a small CRO that we've just started. And again, actually, these systems, the receptors do behave quite similarly. So I think people should be scared of shifting away from GPCR. So there's a lot of work being done here in Nottingham on RTKs. And a lot of that work's been done by Laura Kilpatrick in conjunction with Steve Hill's group. And again, those receptors, you can do a lot of good pharmacology there, and they've developed some really interesting binding assays for looking at interleukin binding on those receptors. And I think again they're all quite similar, really, receptors, I think, in general. So I think we should be scared of moving away from GPCRs into sort of related areas. They are membrane bound targets.
Yeah, but then GPCRs are the best, and everybody should be working on GPCRs
forever. It should be, yeah. I mean, I do agree. You could have another podcast, though.
Yes. Well, I've been trying to talk to people who work on ion channels, with the idea of creating Doctor Channel, but I don't know enough about that. But maybe we should think about Dr. RTK.
It sounds cool, doesn't it?
I think so too. I think so too. I think
I know some good ion channel pharmacology. If you want to get some on the program, there are some really good ones.
That'd be great.
South of working at Sussex University, actually, moved from Novartis. They could probably got some good stories to tell you about some of the drugs that they're developing for certain conditions in the lung.
That'd be great. Yeah, that'd be great. We might need. Yeah, I might need to concede the host part to someone who actually understands ion channels. But I think the other membrane protein communities would benefit from having a podcast and a platform like Dr. GPCR for their own. Well, if anyone's listening, if you want to, we're happy to help set up the Dr. RTK podcast and platform, or Dr. Channel podcast and platform. I think we've had 3 beautiful years of experience, with hopefully many more to come. And I think it's absolutely worth having those conversations. And then we can have a panel with all these experts on membrane proteins and try and figure out which one is the best.
GPCRs.
Obviously. David, thank you so much for your time. I really enjoyed our conversation.
Thank you very much. I'm absolutely delighted to have been on the show, and I've watched a number of these, and I've heard so much from colleagues that I've been around the world with, that I've toured. Yeah, I've toured with Terry a little bit, and Sam Hall was another person you probably know quite well, and they all say very nice things about it.
Thank you. Thank you. I really appreciate it. All right. Talk to you soon. Thank you for joining us and listening to this Dr. GPCR podcast episode. We would like to thank our guest. We also want to say thank you to our Dr. GPCR team members, Attila, Ines, Montserrat, Ivana, Andreina, Balint, and Julia. A huge thank you to our ecosystem partners as well, Domain Therapeutics, GPCR Therapeutics, Design Pharmaceuticals, Montana Molecular, and Orion Biotechnology. You can connect with our partners directly in the ecosystem, so don't forget to join us today. Please subscribe to the Dr. GPCR newsletter, find us on YouTube, and if you like our podcast, leave us a 5-star review on Apple Podcast. You can also leave us a testimonial at drgpcr.com/testimonials. Another great way to support us is to share your favorite Dr. GPCR program with your network and colleagues. With any questions or suggestions, feel free to email us at any time at hello@drgpcr.com. Until next time, stay safe.